In this podcast episode, Andrew Huberman delves into the intricate relationship between hormones, the nervous system, and the regulation of hunger, eating, and satiety. He emphasizes that hormones do not operate in isolation. Rather, they work in conjunction with the nervous system to influence our eating behaviors. Huberman begins by discussing the hypothalamus, a critical brain region involved in hunger and feeding regulation. Within the hypothalamus lies the ventromedial hypothalamus, VMH, which has been the focus of research due to its paradoxical effects on appetite. Disruption of neurons in the VMH can lead to hyperphagia, where animals or humans exhibit excessive eating, or anorexia, where they lose interest in food altogether. This duality suggests that the VMH contains multiple populations of neurons, some promoting feeding and others inhibiting it. Another important brain area mentioned is the insular cortex, which processes sensory information from the mouth, including taste and texture. This region plays a significant role in determining whether we enjoy what we eat and whether we feel satisfied after a meal. Huberman references a classic experiment involving parabiosis, where two rats were surgically connected to share blood supply while maintaining separate brains. When one rat's VMH was lesioned, it became obese, while the other rat lost weight. This experiment highlighted the existence of hormonal signals in the blood that influence hunger and appetite, leading to the exploration of specific endocrine factors that regulate these processes. The discussion transitions to several key hormones that impact hunger and satiety. One of the most notable is ghrelin, a hormone released from the gastrointestinal GI tract that stimulates appetite. Ghrelin secretion is influenced by blood glucose levels. When glucose drops, ghrelin levels rise, signaling hunger. This hormone acts as a kind of biological clock, prompting hunger at regular meal times. Huberman also discusses the role of melanocyte-stimulating hormone, MSH, which reduces appetite. The balance between MSH and the activity of agouti-related peptide, ARP neurons, which stimulate eating, is crucial for understanding hunger regulation. When we haven't eaten for a while, AGRP activity increases, while MSH levels rise after eating. Another important hormone is cholecystokinin, CCK, which is released from the GI tract in response to the presence of fats and proteins. CCK plays a vital role in signaling satiety and reducing hunger. The release of CCK is stimulated by specific nutrients, including omega-3 fatty acids and certain amino acids, which can help maintain a healthy appetite. Huberman highlights the detrimental effects of highly processed foods on hunger regulation. He explains that these foods often contain emulsifiers, which can strip away the mucosal lining of the gut, impairing the gut's ability to detect nutrients and deploy satiety signals. This disruption can lead to overeating, as the body fails to recognize when it has consumed enough. He emphasizes that the consumption of whole foods is beneficial for maintaining a healthy gut and regulating appetite. The negative impact of processed foods on the gut-brain axis is significant, contributing to issues like obesity and diabetes. The podcast also covers the role of insulin in managing blood glucose levels. Insulin is essential for shuttling glucose to cells and maintaining healthy blood sugar levels. Huberman explains the importance of keeping glucose within a specific range, as high levels can damage neurons and lead to various health issues, including diabetes. He discusses the interplay between insulin and glucagon, the latter being responsible for mobilizing energy stores when blood sugar levels are low. This push-pull dynamic is crucial for understanding how the body regulates hunger and energy balance. Huberman provides actionable insights for managing hunger and appetite. He suggests that the order in which we consume macronutrients can significantly influence blood glucose levels. For instance, eating fibrous foods first can blunt the rise in blood sugar that follows carbohydrate consumption. Additionally, engaging in physical activity before or after meals can help stabilize blood sugar levels. He also touches on the benefits of regular meal timing, which can enhance the body's hormonal responses and improve appetite regulation. By establishing a consistent eating schedule, individuals can better anticipate hunger and manage their food intake. Towards the end of the episode, Huberman discusses the effects of caffeine, particularly from mate, yerba mate, on appetite regulation. He notes that mate can increase levels of glucagon-like peptide 1, GLP-1, which acts as an appetite suppressant and helps regulate blood sugar levels. The presence of electrolytes in mate also supports neuronal function, making it a beneficial beverage for those looking to manage their hunger and energy levels.